This is the third video on Key Area 2.1 and it's looking at regulating the activity of enzymes. So first of all, I want to have a wee look at the direction of enzyme action. Now, a metabolic pathway we've seen already in this example here, this metabolic pathway going from metabolite W to metabolite X to metabolite Y to metabolite Z are controlled by enzymes. Now, as this metabolite here becomes available, what will happen is that enzyme 1 is going to become active, and as X is formed, enzyme 2 is going to become active in converting metabolite X to metabolite Y, and so on. As metabolite Y becomes available, enzyme 3 will become active. So a continuous supply of metabolite W will ensure that the direction of the enzyme sequence will go this way from W to X and then so on to Z. So this supply of W entering the system drives the sequence in the direction W to Z. But what happens if, say, maybe you get too much of metabolite Y, it builds up and you get too much of it? Well, most reactions are reversible. I did say in the last video some reactions aren't reversible, but most of them are reversible. So if you get this situation here where you get too much of this metabolite Y built up, then it's possible for an enzyme to convert metabolite Z back to metabolite Y in order for everything to become balanced again. Now, what I want you to think about as well is that every single one of these enzymes here are produced by a gene, and because each enzyme is coded for a gene, if the gene is expressed, the enzyme is formed. Now, if you've got a situation like this, then what can happen is that the control of these genes can actually control the enzyme activity. So what we're going to be looking at are some pathways that only need to operate in certain circumstances. And to prevent enzymes being produced when they're not needed, genes can be switched off or switched on. An example of that is looking at something called the lac operin in E. coli. Now, E. coli are a bacteria, and we're going to be looking at a pathway here that involves the breakdown of lactose, which is sugar in milk, to galactose. Now, there's an enzyme involved in that, and that enzyme is called beta-galactosidase. Now, if there's no milk present, there's no lactose present, then there's no point in the E. coli producing beta-galactosidase. And it is controlled by this operin. Now, it's called the lac operin because it's talking about lactose, but an operin basically is where you've got one or more structural genes are called, and here's a structural gene here. And you've got a neighbouring operator gene which controls this structural gene. And the operator in turn is affected by a repressor molecule. So here's our repressor molecule here. And that repressor molecule is coded for by a regulator gene. So this is the operin, and in this situation here we're looking at the lac operin. So in a situation where there's no lactose present, what happens is that this regulator gene that is here, transcription and translation take place in that regulator gene to form a repressor molecule. And that repressor molecule combines with this operator gene here. And when it does that, the operator gene switches off the structural gene. Now, the structural gene is the gene that codes for the enzyme beta-galactosidase. So if that gene is switched off, then no beta-galactosidase is produced. That's in the absence of lactose. But if we look at this, if lactose is present, then what happens is that you've still got, in this situation here, you've still got your regulator gene, you've got this repressor protein molecule, you've still got the repressor molecule produced. But in this situation here, lactose enters into the cell, and what happens is the lactose combines with the repressor molecule. So here it is over here combining with the repressor molecule. Now because the lactose is combined with that, it's not, the repressor molecule is no longer able to connect up here to the operator gene. So the operator is left repressor molecule free. And what happens is the operator gene will switch on the structural gene here. And so you get this gene switched on, so you get transcription and translation to form the enzyme beta-galactosidase. Now beta-galactosidase will then digest the lactose, breaking it up to galactose until the supply runs out. And in that situation, the lactose is no longer able to bind to this repressor molecule because there's not enough of it there, the supplies run out. So the repressor molecule then attaches to the operator gene, and the operator gene switches the structural gene off. So this is a form of controlling the enzyme activity here. The gene is switched off when the enzyme is not needed, and the gene is switched on when the enzyme is needed. In this situation here, we call the lactose a signal molecule because it's a signal really of the start of this process of the beta-galactosidase being produced. So we're looking at this control of this enzyme here taking place at the gene level here. But there are some pathways in which the genes are always switched on and the enzymes are always present. So how can these pathways be controlled? Well, one way of doing this is this same sort of situation here with signal molecules, but we're not talking here about these signal molecules switching genes on and off. What we're doing is we're talking about a molecule that acts as a trigger to set off events that activate enzymes. And an example of that is adrenaline. Now adrenaline is released in the adrenals, and what happens is it travels through the bloodstream and binds to the membrane of liver cells, and there it triggers a series of events that activates the enzyme that converts glycogen to glucose. In this situation here, the signal molecule comes from other cells in the organism's body. So what we're saying here is that the liver cells, there's a pathway there that is being controlled, but the signal molecule is produced in the adrenals and other cells in the organism's body, and they travel there via the bloodstream. There are signal molecules that affect a cell's metabolism, and they can originate in the cells themselves. So there may be, for example, in the liver, cells that control, signal molecules that control other pathways that are there in the liver cells, and they're produced within the liver cell themselves. But adrenaline is produced elsewhere in the body. So that is signal molecules. And other things that can affect enzyme action are inhibitors. An inhibitor is something that decreases the rate of an enzyme controlled reaction. The first type of inhibitor we're going to have a look at is a competitive inhibitor, and it works by having a similar shape to the substrate molecule. Now that means they can fit on the active site. They remain unreacted because they have a different structure to the substrate, but what it means is that if that competitive inhibitor is binding to the active site, then there's less substrate molecules that are able to bind to the active site, so the reaction rate is decreased. So let's have a wee look at what we're talking about here. Here's an example up here of just a straightforward reaction taking place. There's no inhibitor present. You've got your substrate, binds onto our active site, and you have to think about the induced fit situation, things there as well, but that's not in this diagram here. We've talked about it before. So we've got the enzyme binding to the substrate, and then we've got the situation where the enzyme then releases the product. So there's no inhibition in this. But in this situation down here, down below here, there is an inhibitor. And what we're saying here is that here's our substrate here, but the inhibitor is able to bind onto the active site. And because it binds onto the active site, it prevents the true substrate binding to it. So it inhibits the enzyme. Now when a reaction is taking place, you're thinking about there being lots of enzymes that are present there, and you'll have substrate present. So I'll try and draw the substrate. You'll have some substrate present, and you'll have some of the inhibitor present. I'll just draw another one of the inhibitor. Now we say that competitive inhibition is reversible, and it can be reversed, and it can be reversed by increasing the substrate concentration, that is adding more substrate. So if I add more substrate, more and more substrate, I'm not very good at drawing, as you know, then it increases the chance of a substrate molecule colliding with the active site. So the more substrate there are, then the more likely that substrate is to bind with the active site, and it prevents the effect of the inhibitor, or it overcomes the inhibition. So we can reverse this process by increasing substrate concentration. And we'll have a look at a graph at the end of this that just shows this effect that's happening as well. So because we've got competitive inhibition, it makes sense to think that there's such a thing called non-competitive inhibition. Now in a non-competitive inhibitor, it doesn't bind to the active site. It binds to some other place on the enzyme, and it causes a conformational change in the enzyme. So what we're saying here is, if we have a wee look at this diagram over here, I'll just move it over just a little bit. Here's our ordinary reaction again up here, but our inhibitor here binds to another area on the enzyme, and when that happens, it changes the active site, and you'll see here's the change in the active site. Now because it's nothing to do with the substrate competing with another inhibitor that is going to join on to the active site, increasing the substrate concentration isn't going to make any difference, because at the end of the day the active site has been changed, and therefore the substrate cannot bind to the active site anyway. So it's not the same situation as you had with the competitive inhibition. So increasing more substrate will not make any difference to this sort of inhibition. This type of inhibition can be reversed in that this inhibitor here can be removed, but sometimes it's irreversible depending on what the inhibitor is. And I've just put on here the change in the 3D structure of the enzyme means it can no longer catalyse the reaction. So since they don't compete with the substrate molecules, non-competitive inhibitors are not affected by substrate concentration. So here's a graph just looking at these ideas where we're increasing the substrate concentration down here. Now, this is an uninhibited reaction, so there's no inhibitors about, competitive or otherwise. And the reaction takes place as we increase the substrate concentration. You'll remember this from the last video. The reaction rate will increase until it reaches a point here where it can no longer increase anymore because all the active sites have been used up. When you're looking at, if you're graphing out a competitive inhibitor, then what happens is you get a very similar curve, but it's lower down. The reaction rate is lower. And the reason for that is because, remember, you've got some of these inhibitors that are binding to the active site. And depending on how much substrate is there, if there's more substrate present, then more of the substrate will be able to get in there before the inhibitor does. But if you've got less substrate, say a situation down here, then you've got more inhibitor present in the substrate, and so therefore what happens is you have more of the inhibitor able to bind to the active site, and so you get more inhibition happening down at this point here than you do up here where there's more substrate available. But you do eventually reach a point where, again, the active sites are full and the reaction rate can't get any further. Now, when we have a wee look at the situation where there's a non-competitive inhibitor, what's happening in this scenario here is that we've got our enzyme and the inhibitor is binding to that and changing the shape of the active site. And so it doesn't matter how much extra substrate you're adding, the reaction rate will not get any higher. So what's happening here is that more and more of the enzymes are getting more and more of the inhibitor added to them, and it's changing the active site. Initially at the start of this here, then there's some of the enzyme that's not got the inhibitor attached to it, and so you do get a certain amount of rate of reaction until you get to the point where all the enzymes have had the inhibitor attached to them and the active sites are all affected. So you can add substrate to your heart's delight and it's not going to make any difference at all to the effect of the rate of reaction. Now we're going to have a look here at this here called allosteric or feedback inhibition. Now some enzymes involved in metabolic pathways are controlled by a self-regulating mechanism. There are some substances, and usually it's the product, or often it's the product itself that acts as an inhibitor. And the inhibitor binds to the enzyme at a specific site and modifies the active site of the enzyme, and that prevents the binding of the substrate molecule. And the sites on the enzyme where this happens are called allosteric sites. And these type of enzymes are called, the general term for them is allosteric enzymes. So looking at a situation here, we're looking here at a feedback inhibition situation here based on just what I've been talking about there, where an enzyme, a product can bind to an enzyme and inhibit it from inhibit its reaction. And because we're talking about an end product here doing that, we'll call it end product inhibition. And in this example here, the amino acid isoleucine is made through a conversion of 309 in a 5 enzyme controlled steps in a bacteria. So here we've got our initial substrate and we've got our enzymes that are involved in breaking that down until we get this here formed, the isoleucine. Now what happens is, as it builds up, the isoleucine actually binds to an area on this enzyme here, on enzyme 1. And just like I've been talking about before, where you've got this allosteric site. So and what it does is it binds to the enzyme and inhibits it. And that inhibits any further production of the isoleucine. And by doing that, the cell only produces the amount of the product that it needs. Again, I'll post another video on the iTunes U that goes over some of these points as well.